Hi, how are you?
Good. How are you?
Good. Can you remind me how to say your last
Ranghini.
name?
Yeah, Ranghini. But Egon Ranghini. It's fine.
Well, should know how to say your
Yeah, I know it's
So
fine,
never
but
all
it's
these
it's the hard with the hard G or
Okay.
the
Egon Ranghini.
yes, perfect.
Egon Ranghini.
Perfect.
Thank you. So how was the travels getting here today?
Yesterday, so we actually drove here yesterday from Madison, from Milwaukee.
Okay.
So that was fine. It was raining. It was raining.
I was thinking of this morning when I was coming in. I'm like, I wonder if they're flying in now or blah, blah, blah. I just have to come around the lake. So by the time I came around the lake,
There's
It's all
nothing.
yeah, it's all right. And we just really got heavy wet snow. It's not slick, which is,
Yeah, it's not all ice.
yeah. Hello, everyone. I'd like to welcome you to this week's UW Biotech Talk here in the Biotechnology Center. These talks happen monthly on the Thursday at the third Thursday of the month at 1:30 p.m. This week, we have 10x Genomics here joining us, and they are going to give a talk on time and flexibility are on your side with single cell gene expression flex. The UWBC Gene Expression Center offers a 10x genomics service for both single cell, single nuclei, and now as of January 3rd, 2023, we will also be providing the fixed RNA assays as well for human and mouse that are currently available through 10x Genomics. In addition, beginning the first of the year, the Biotechnology Center will have the Chromium controller available for independent access in the Biotechnology Garage on the second floor of the Biotechnology building. I'd like to clarify this Chromium controller that will be available for independent access after you've been satisfactorily trained by biotech staff will only be able to do, will not be able to do the flex assays that will be discussed today in today's talk, okay? So that pretty much just entails the current assays, excluding the fixed RNA assays that will be able to be done on the Chromium controller in the biotech garage. If you are interested in learning more about that, please send an email to gcinfo@biotech.wisc.edu indicating your interest. In addition, there will be a formal announcement in the UWBC newsletter that goes out monthly. I don't believe it's gone out this month, but it should be going out shortly, and there will be an announcement about the independent access for that instrumentation. So without any further ado, I'd like to introduce Egon Ranghini, who is going to give our talk today on this assay. Thank you.
Thank you, Sandra. Thanks a lot. Well, good afternoon, everyone. Thanks for joining us. Thanks for taking the time to meet with us today. My name is Egon Ranghini. I'm one of the science and technology advisor for Tenex. And and today, so I'd like to focus on the new solution, which is called single cell gene expression flex. Is essentially a solution that allows you to fix samples in formaldehyde and then store the samples and run them even months later. So it really gives you a lot of flexibility, no pun intended, I guess. Gonna talk about that. And I will also cover a couple of more things, just a couple of things at the very beginning. And one more thing, I'd be more than happy to share this presentation with you. And if you have any question, feel free to ask me or add your question in the chat, and make sure that we can answer your question. So that's just to show you the three main platforms that we have available at Tenex and what we've been developing to puzzle the complexity of biology. So today we're going to focus on the Chromium platform, or at least a couple of solutions on the Chromium platform that is for single-cell genomics. But keep in mind, again, that we have our spatial transcriptomics platform called Visium, and then is for all transcriptome analysis in FFPE or fresh frozen tissue sections. And last week, we started shipping the new in situ analysis platform that is called Xenium. And that will allow you to interrogate the expression level of hundreds of target genes in FFPE or fresh frozen tissue sections and achieve subcellular resolution. Now, back to single cell. I always like to say that we are extremely proud to see that this technology has been used in more than 3,700 publications, and is at the same time really super excited to witness the research that has been made possible thanks to Tenex. Now, when we look at a Tenex workflow, single cell, or pretty much any workflow, we should say, we always look at what we call a beginning to end solution, beginning to end workflow, because all the reagents that you need to make a sequencing ready library, those reagents are already provided in the kit that we sell. And so when you start a single cell experiment, all you need to do is to prepare a high quality single cell suspension or single nucleus suspension if you are profiling nuclei. And then we also have a pretty broad range of, let's say, bioinformatics tools to essentially run data analysis and to visualize the data. And those tools are available for you for free. I would also like to emphasize that we have a cloud analysis solution that allows you to essentially run our single cell pipeline for free using our clouds. You don't need any bioinformatics expertise, as is a GUI interface. So really you need to, all you need to do is to create an account with us, and you'll be able to run Cell Ranger in a matter of hours. Now, when we consider those 3,700 plus publications, now we know that the majority of those papers have been generated using human or mouse samples, but we do also have solutions that we also have a lot of papers that have been published or generated using non-traditional model organisms, and that ranges from plants to insects to non-human primates to fishes. Not sure about elephants here or octopus, but maybe one day. And when it comes to sample type compatibility, here is where really we can profile a large variety, or we can start rather from a large variety of sample types. And so We know we can start from single cell or cell in culture. We can start from single nuclei or isolate nuclei. We can start from blood samples and isolate the cells. We can start from fresh tissue or snap frozen tissue. We can now also fix tissue in PFA, in formaldehyde. This is actually what I'm going to talk about today in more details. And then we have this very exciting and new protocol that will enable us to essentially generate single cell data starting from FFPE tissue blocks. It's super exciting, and I'm going to show you some slides later. But before we get there, I just want to give you an example of how the technology has been used in the field. And I chose this recent paper, which was published in Nature Neuroscience a couple of months ago. And it's a study in which the authors wanted to kind of better characterize this condition called frontotemporal dementia. And so to do that, they obtained brain samples from patients with frontotemporal dementia, and they profiled more than 430,000 nuclei using the single-cell gene expression solution. In a nutshell, they were able to identify a subpopulation of astrocytes that are called GM, that stands for gray matter astrocyte, that were particularly enriched in the patient compared to controls, and the authors found that those astrocytes might be associated with the dysfunction of the blood-brain barrier. And so this is a really nice example that shows you how you can use a single-cell approach to interrogate the heterogeneity of a very complex samples and really start getting very impactful and meaningful information out of a complex sample such as those brain tissues. Now, in this paper, the authors profiled nuclei. We know that isolating nuclei from tissue can be challenging and very time-consuming. And so few months ago, we released a nuclei isolation kit, which is compatible with a large variety of tissue types, frozen tissue. In particular, it also works with fresh tissue. And all you need to do here is use 3 to 50 milligrams of tissue and have a benchtop centrifuge. And in about 1 hour, you will isolate high-quality nuclei that can be pretty much immediately run on our assay. Really, this kit is intended to generate high-quality nuclei with no optimization or very little optimization. And everything that you need for nuclei isolation is part of the kit, including the RNase inhibitor that's already included in the kit. And so to show you, for instance, how we use this kit in-house, here we looked at 4 different tumor samples. So we isolated nuclei from fresh frozen, or snap frozen, I should say, tissues. And so we have an ovarian cancer tumor here, ovarian cancer here, we have a kidney cancer here, melanoma, and also we have a pancreatic cancer here. And as you can see, those 4 different types of cancer really separate well or cluster well apart from each other. And we were also able to retain the immune cells, and so not only the cancer type specific cells, but also the immune cell population is very well represented. And here we have another example, which we use nuclei as input material for our multiome assay, which allows you to simultaneously measure gene expression and profile the region of accessible chromatin. And again, we isolated nuclei from a kidney tumor sample. This was a human sample, and we immediately loaded the nuclei on the instrument, generated the data, and we were able to identify the main components of the kidney. But what I would like to show you, what I think is really important here, is we were able to retain a large percentage of podocytes. And I'm a kidney guy, so I really love to see this data, because we know that podocytes are really large cells, and so sometimes it's challenging to capture those cells. But now that we have nuclei, the cell size is no longer an issue, so we can really have a beautiful representation of those cell types. Now, very briefly, the kit also works on fresh tissue. So here we see a sample. It was a spleen sample, I believe, fresh and frozen. There is a very good overlap between the 2 clusters. My recommendation is, if you want to run fresh tissue, test the kit first. And the the differences that we see in the cell population between the two conditions, those are essentially due to the fact that some cell types don't like being frozen. And so that's also something to keep in mind when you plan your experiment. If you want to retain a particular class of cell, like let's say neutrophils, that would be better. It would be better if you use fresh tissue instead of frozen tissue. And a question that you might be asking is, well, okay, I'm profiling nuclei instead of whole cells. What type of information I'm missing? Well, the truth is, for the many samples, the vast majority of samples, I should say, you don't lose any information, actually. And that's because, true, you capture less RNA, and so you will see less genes per cell, for instance, but the biological information is retained. And so you will still be able to identify the same population of cells with pretty much the same proportions. And this is PBMC data, but there are also papers done on cancer, so even more complex samples. Now, we know that working with fresh tissue, fresh cells also present many challenges, and this slide doesn't necessarily represent all possible situations, but I think it does a good job summarizing the three most common scenarios. And so we know that, for instance, you might have samples available outside the regular business hours, or you might want to, let's say, collect samples over multiple time points because you're running a longitudinal study, and then ideally you want to submit those samples to at the same time to your genomics core. Or definitely there are tissue cells that might degrade very quickly, and so what we want to do is to preserve or block the degradation, preserve the biology, block the degradation as soon as possible. And then there could be logistic constraints. So, for instance, again, you're submitting samples to your core, or you're receiving samples from a collaborator. And so to tackle those challenges and provide a better solutions, recently we launched a kit which was called the single cell fixed RNA profiling. Now it's called single cell gene expression flex. So probably mentioning the name Flex. And as Sandra mentioned, Flex is not compatible with the Chromium controller. It is compatible with the Chromium X series instrument. So you will need to work with the core when running this assay. So what you can do with Flex is to collect your samples. Then you can fix your samples in 4% formaldehyde plus couple of components we sell with the kit, but 4% formaldehyde is the final concentration for the fixative. And then you can store your single cell suspension or single nucleus suspension for up to 6 months at -80 degrees. So that basically you can preserve the biological state of your samples, really, no matter when you collect them or where you collect them from. And so that's really what gives you a lot of flexibility. And for this particular solution, we use a probe-based chemistry, in which we use essentially, we have more than 55,000 set of probe pairs, or probe pairs, I should say. Those probes have been designed to detect all annotated isoform of a particular genes, and in total, we detect more than 18,000 coding genes. So that's essentially will cover the entire transcriptome, minus some genes that we didn't include because really we didn't add anything or any useful information. Now, because of the probe-based strategy, what we see is a significantly higher sensitivity with this kit compared to the traditional 3' chemistry or the 5' chemistry. Actually, the data is pretty much the same. And then because of probe-based chemistry, we can now also reduce the minimum recommended sequencing depth to just 10,000 read pairs per cell. So when you sequence those libraries, you will be able to save some money also on the sequencing side. And what I'm showing you here, those are not cherry-picked data. We see this trend across pretty much all samples that we tested. And now this trend also has been confirmed in the field by a couple of laboratories, I would say. So this is one, and it's a bioarchive paper from Luciano Martellotto and Jasmine Plummer. Jasmine Plummer used to be a Cedars-Sinai. Now she is a St. Jude. And then so in this manuscript, or I should say in the first part of the manuscript, those authors compare the flex chemistry against the 3' chemistry on the same sample. And they found that the flex chemistry could detect more than twice the number of transcripts compared to 3'. And importantly, they also had a better detection of lowly expressed genes. And as an example, they showed the expression of the top 19 transcription factors. So that is really, you know, the advantage. You're basically now, if you use Flex, you're using by far the most sensitive chemistry that we have available. We have also similar data on 5', and actually this is 5' data on nuclei isolated from frozen tissue. The nuclei were isolated using the nuclei isolation kit, and nuclei were fixed and then profiled with Flex. And again, as you see here, there is a significant increase in sensitivity compared to the 5' chemistry. And in terms of the data quality, now if we look at the fraction reading cell, which essentially measure the background of the assay, again, we get a higher fraction reading cells with Flex, which shows you, again, a better data quality overall. Now, the last piece of data I want to show you is a comparison that an independent lab just generated, and they compared our chemistry against the PARS Evercode V2 chemistry. And again, they found the same sample, so this was human PBMCs. They found a significantly higher sensitivity with Flex compared to the V2 chemistry from PARS. And again, so that's to say, with Flex, really you have an unmatched sensitivity, so you can get more genes, get more data, better understanding of biology, and that is achieved at a significantly lower sequencing depth. Now, in terms of sample types, as it was mentioned before, because this is probe-based chemistry, we have set of probes, and those are available for a human or mouse sample. Now, when it comes to the sample type compatibility, here is where really we can fix a lot of different sample types. And to be honest with you, I haven't found a sample type, at least in the mammalian realm, that we can fix. Probably there is something we cannot fix. If you find something, please let me know. But we have protocols for the protocols that we can use to fix cells, nuclei. We can fix tissue, fresh or frozen. I have a slide later that I can show you, but essentially here what you'll be doing is to collect your tissue, chop or mince your tissue, fix it in the fixative, in the fixation buffer, and then that tissue can be stored at 4 degrees for 7 days. Then you'll need to isolate a single cell. Once you isolate the cells, those cells can be stored for months, again at -80 degrees. So it really gives you a lot of flexibility. And then we can, I'll show you later, but we have a protocols to do single cell RNA sequencing starting from FFPE blocks. And so one important thing that we wanted to make sure is that, well, now we fix tissue or we fix cells, we leave the cells at -80 degrees for, let's say, up to 6 months. How does the cell population ratio change if it changes? And well, actually it doesn't. So really, regardless of the length of fixation or the temperature, the relative proportion of cell population is not impacted. Now, here what I would say is it's recommended to store samples at -80. That is better than storing samples at -20. So if you can choose, always store samples at -80. And in terms of the tissue, so here we took a human colon tissue sample, we fixed it, so we chopped it, we fixed it, and then we isolated the single cells. And as you can see here, we were able to identify different classes of immune cells, and then colon-specific cell types, such as the goblet cell that we have here, some myofibroblasts, a tiny population, or maybe actually to be more here, or enteric, tiny population of enteric neural crest cells, and mast cells. This is a very tiny population here. Now, the Flex solution is also a multi-omic solution in the sense that you can simultaneously measure gene expression and profile the expression level of cell surface protein by labeling cells with oligoconjugated antibodies from BioLegend. Is the total-seq B, as in Bravo. antibodies. Couple of things that you need to keep in mind. One is you will need to you need to label the cells after fixing them. Sorry, the other way around. You need to label the cell before fixing them. So you first label the cell and then you fix them. And then samples will need to be run in a single plex format, not in a multiplex format. I'll talk about multiplex in a second. Before showing you that, so we're also working on a protocol that will enable intracellular staining. That is not available yet, but my understanding is that it will be available fairly soon. And I'll make sure that I'll communicate this information when this protocol is going to be available. Now, multiplexing, this is the other, or one of the other great things about this solution now. So we can multiplex samples and run multiple samples in the same channel of the microfluidic chip without labeling cells with oligoconjugated antibodies or oligoconjugated lipid molecules. So in a typical cell hashing experiment, you'll need to label cells with oligo-tagged antibodies and then combine them. Well, here we don't need to do anything of that. That's because we included a unique barcode to each set of probes. And by leveraging the information of this barcode, during data analysis, we can demultiplex these samples. And so here the advantages are, first of all, we don't need to buy antibodies anymore. Second, we don't need to decide upfront which sample we want to pull together. With Flex, you simply pull together the samples in the way that makes most sense for your experiment, not when you start the workflow, but when you not when you collect your samples, but when you start the workflow. So technically, you have up to 6 months to decide whether or not you want to combine samples. And so that's the other great advantage. And when you do combine samples, you can pull together up to 16 different samples in the same channel and target up to 120,000 cells per channel. A chip here has 8 channels, and so a full chip will allow you to recover 1,024,000 cells per run. And of course, when you multiplex samples, so is where you also we're going to start saving money. I'll show you a slide later anyway. Now, here we want to show you that when you multiplex sample, essentially there is no batch effect due to multiplexing. We profiled 112,000 cells from a mouse spleen, and we split the initial pool into 16 different initial samples into 16 different pools. And again, as you can see here, the UMAP projection are essentially identical, and so really there is no batch effect. And in here, we run this easy assay to essentially provide an idea again of how this kit can be flexible and and the sensitivity of this kit. So I'm not going to show you any groundbreaking biology because this is a very well characterized assay. And so it's an assay in which you will be activating T cells by incubating resting T cells with Dynabeads conjugated with anti-CD28 and anti-CD3 antibodies. So you do that, and resting T cells become activated. So we had 2 independent donors, PBMC donors. We stimulated these cells for up to 2 hours, and then included 2 controls, a T0 and a T equal 2 hours. And so we had a total of 16 samples, and we were able to pull those samples together and run them in just one channel. And then we looked at marker for activation, T cell activation. So we started with FOS. There is pretty much no expression in the controls, but then the expression already is already detectable after 5 minutes after the after the stimulation, and it peaks around 30 to 60 minutes, I guess. CD69 is another marker for T cell, early marker for T cell activation. The expression is very low in the controls, and then it peaks around 60 to 90 minutes as we would be expecting. And finally, VISTA, this is a immune checkpoint marker. Expression peaks around 60 to 90 minutes, and the expression is very low as expected in the control. So again, I'm not showing you any groundbreaking biology. This is a very well characterized assay, but I'm hoping that this gives you an idea of what you can do with this kit if you are collecting samples over multiple time points, Now you can collect them, you can fix them, and essentially you don't, once cells or samples are fixed, you don't worry anymore about cell viability because samples are being fixed, are being stabilized, and so we don't need to worry about keeping samples alive anymore. So this is a slide that perhaps gives you an idea of what you can do with the different samples. Again, and we have protocols, so you don't need to develop, optimize any fixation protocol or protocol for fixation. So if you start with cells or nuclei, you can fix them, store them, and then store them at -80 for up to 6 months. If you start with tissue, so now we can decide whether maybe we want to dissociate the tissue into single cell. Let's say maybe you want to do FACS, and then we can fix those cells, store them. We can isolate nuclei using nuclei isolation kit, fix the nuclei, store the nuclei, or we can chop mince the tissue, fix the tissue, store it for 7 days, 4 degrees, and then isolate cells afterward and store those cells. And if we have frozen tissue, again, you can decide to isolate nuclei, fix the nuclei, store them, or you can chop mince the tissue, fix the tissue, store it, isolate the cells. So it's really a lot of flexibility that now you have. Now, there are a couple of considerations to keep in mind when we work with fixed samples. First of all is the number of cells or nuclei that we want to start with, because there are some washing steps after fixation and after the hybridization step. It is recommended to start with at least 300,000 cells or 500,000 nuclei. If you start with tissue, it's a good idea to start with 25 milligrams of tissue. Sample dissociation, if you do dissociate the tissue and you want to keep the tissue alive, let's say isolate single cells, high viability is critical for in order to get high quality data. But if you fix the tissue, well, that's no longer a concern. Tissue is fixed, and so we don't worry anymore about viability. Now, when you fix the tissue, I mentioned the fixative is final concentration is 4% formaldehyde. You will also need to use a sample prep kit that we provide. It's very inexpensive, but it contains a couple of components that you need to add to the fixation buffer. So it's not just 4% formaldehyde, it's that plus what we provide with the kit. And when you count your cells post-fixation, strongly recommended to use fluorescent. That's because if you do use trypan blue and you have some debris, you might have some false positives. So the trypan blue might see debris as a cells, and so your cell count might be off. So really strongly recommended to use, for instance, actinium homodimer 1 when counting fixed samples. Now, to this protocol here, again, I keep saying it is super exciting because it's almost incredible when we can run single cell starting from an FFPE blocks, but we can. In a nutshell, and the protocol is available. So if you scan this QR code here, you can download the protocol or just send me an email and I'll send you the protocol. So in a nutshell, what you'll be doing is to cut a couple of curls or scrolls of your FFPE blocks, and then dissociate the tissue, isolate single cells, and profile the cell with the flex chemistry. We have now 2 different tissue dissociation protocol. One uses a GentleMax, and so you will need to have access to that machine. Plus, you also need to use Miltenyi reagents. There are a couple of there is a kit that will need to be used. But then now we also have a manual dissociation protocol that does not require the GentleMax. This protocol was actually released last week, and I've been told that it's more robust than using the GentleMax. So if you are starting right now, my recommendation is to use the manual dissociation protocol. Now, this data here was generating using the Miltenyi kit because it was released before we actually released the manual protocol, but it shows you that we cut 2 scrolls, 50 micron each, so it's not much, out of the FFPE blocks. This is a lung cancer sample, and we were able to identify different classes of immune cells, again, lung-specific cells and those cancer cells here. I just want to show you an example of what happens if things don't go really well. So here you have a human pancreas samples. again, it's really nice to see good quality data from pancreas. It's a very RNA-rich tissue. If you collect it, fix it immediately, or embed it immediately, in this case, in FFPE, then you can really get beautiful data. This is an example of a poor sample, what we call failed samples. I would not focus too much on the confidently mapped reading cell. The difference is not staggering here, but the library complexity is. So here we detect only not even 100 genes per cell, not 100 UMI per cell. And as you can see here, we don't really get a nice separation of those different cell populations, what I would call a bouquet, rather than a cluster separation. And so that is what, if a sample fails, that what might happen. But so far we tested about 30 samples, 30 different blocks, and more samples have been tested as we speak. You might also wonder what happened when we compare the FFPE protocol with the CHOP means, the regular protocol. Remarkably, those two protocols yield very similar data. And I say remarkably because getting good quality data from an FFPE blocks can be, I mean, it's almost incredible. So again, you will get either way good quality data. It's also nice to see that this chemistry has been proven successful in the field. This is the same bioRxiv paper from Luciano Martellotto and Jasmine Plummer that I mentioned before. In the second part of the study, they generated a protocol to isolate nuclei from FFPE blocks. And I love the quote from Luciano here, where he say, well, a tweet, I should say, where he said that the cure for cancer is now sitting in a pathologist drawer. And so what they did was to isolate, develop this protocol to isolate nuclei from FFPE blocks. And then they profiled the nuclei using the flex chemistry, and they also used a consecutive section that was profiled using Visium for FFPE solution. And they found a very nice correlation in terms of gene expression, in terms of cells detected between the two technologies. And so, paraphrasing Luciano in my last bullet point, what I would say is this protocol, or our protocol, now really enables you to generate high quality and high sensitive single cell or single nuclei data starting from FFPE tissues, which essentially now allows us to generate extensive retrospective studies using our archived sample. And this essentially gives us the unique opportunity to do single cell or single nuclei and spatial transcriptomics starting from the very same tissue block, which gives us really an unprecedented view of biology, and nobody else can do that. And so, for instance, Luciano here, they integrated their single nuclei data with their spatial transcriptomics data. And by doing that, that's a strategy to increase the resolution of the spatial transcriptomics platform. But they were able to see essentially that the epithelial markers here commonly expressed in the metastasis tissue, those overlap with the metastasis region of the tissue. Hepatocyte markers, those overlap well with the healthy part of the tissue. And then they found ESR1 expression. And that is really interesting because this patient was originally a ESR1 positive, but then following therapy, she was she became ESR1 negative. And this sample was supposed to be ESR1 negative, at least when they obtained this sample, that was supposed to be a triple negative cancer. And so ESR1 was not supposed to be expressed, but ESR1 was detected with the single cell data and also with the spatial transcriptomics data, which shows you again the incredible sensitivity of this technology, specificity, and also shows you that the typical diagnostic method cannot really provide that level of sensitivity. I leave you with a paper or manuscript that we recently released. And it's an example of how we use the entire 10x ecosystem to profile the same FFPE tissue block. So this was a human breast cancer sample. We cut a couple of scrolls and generated single cell data using Flex and the Chromium X. Then we took a consecutive section and generated spatial transcriptomics data using the CytAssist. And then again, a consecutive section and generated in situ data using the Xenium analyzer and generating in situ data. And that is really what we say, well, this is the ecosystem that really gives you an unprecedented view of biology, because now you can use 3 different approaches, 3 different platforms to study just the very same tissue block, the same tissue sample. And I really recommend reading it. Now, in the last 2 minutes, I wanted to briefly mention the cost saving, or how much this solution costs. And keep in mind that what I'm showing you here are just 10x genomics, is just the cost for 10x genomics reagent. So what I'm showing you here does not include any core fees or any sequencing or the cost for sequencing. It's just the cost of the kit. Okay. And as a benchmark, I'm using the 3' chemistry, the typical 3' chemistry. So the cost per sample is about $1,600 with the 3' chemistry, a little bit more, and it's going to go up starting January 1st. A little bit up. It's going to go a little bit up starting January 1st. Now, this is Flex. And with Flex, we have 3 sites of the kit. The first one is what we call a 4x1. Here is where you have 4 gel beads reaction and only 1 set of barcoded probes. So you will not be able to multiplex samples. This is the kit that you'll be using to run essentially 4 samples separately, 1 sample per lane. It costs $700. Well, it's about $700 $1,700 per sample. The price is not going to go up. Flex is not going to go up. And so yes, it's a little bit expensive. This difference will be a little bit lower because 3' is going to go up, but it's a little bit expensive, more expensive than 3'. But keep in mind the increased sensitivity and increased flexibility with Flex. Then we have what we called a 4x4. So 4 gel beads, 4 sets of barcode. That allows you to multiplex up to 4 different samples together in the same reaction. Target 10,000 cells per sample, so there will be a total of 40,000 cells per gel bead reactions. And in total is essentially a 16 reaction kit, pretty much. So now you see that when you run this kit and you combine 4 samples together, the cost per sample is about $1,300. So now you're starting saving about $300 compared to the 3' or the 5' chemistry. The price is the same. And then we have the 4x16. This is where you can, so 4 gel beads, 16 barcodes. This is where you can combine up to 16 samples together. And it's a total of 64 reactions. Now, you see that here, when you do take advantage of full advantage of the kit and combine 16 samples, essentially each sample will cost you $550. And quoting one of these sequencing core directors in my region, they basically mentioned that at this point, $550 per reaction is pretty much what we used to pay for bulk RNA sequencing just a couple of years ago. Again, this is just 10x genomics reagent cost. It doesn't include any other, it doesn't include sequencing, it doesn't include core fees. There is even a larger size of the kit in which we have 16 gel beads and 16 barcodes, so a total of 256 reactions. That will reduce the cost per sample down to $380, 280 and change. I believe it's $280, $285 per sample. So we can really go down in terms of cost per sample. And so I'm hoping I was able to show you that we are kind of at an inflection point now where I strongly believe that Flex will become the new standard, the new gold standard for single-cell RNA sequencing. And that's because of this superior sensitivity, its flexibility. Again, we can run anything between 1 sample and 128 samples per chip, or up to 1 million cells per run. The type of sample we can fix, again, we can fix many different types of samples. We can store them, and we don't need to worry about cell viability anymore once samples are fixed. That gives us a lot of peace of mind and flexibility. When you do multiplex sample is where you'll be able really to save money and save time, and also reduce any potential batch effect, maximize your sample, the way you want to perhaps run biological replicates, for instance. And then it's really the only chemistry that you can use to do RNA sequencing starting from FFPE samples. And so in conclusion, what I would say is that our goal here is really to provide you with an ecosystem that enables you to do fantastic research. We have been spending a lot of time and effort to make sure that those three platforms are very well integrated together and can work on as many sample types as possible. We have also incredible resources that really are there to help you and make sure you're going to be successful. And so we really want to we'll start having a conversation with you, starting even here before sample prep, when you start thinking about designing your experiment, setting up your project, and then we can go through sample prep all the way down to data analysis. We have a resource for all of that. So what I want to say is our goal is really to make sure you're going to be successful. So don't hesitate to reach out at any time. We are really here to help you. I'll leave you with this slide, and yeah, thank you for your attention. I'll be happy to take any question, and happy holidays if we don't see each other again before the end of the year. Thank you. Yes.
Egon, I have a question. So at the end of November, Genomics announced that a partnership with 10x Genomics.
Oh.
The CRISPR clean assay, which they are advertising being compatible with the 3' single cell RNA assay. Will that also be compatible with Flex? And what does this partnership really mean between Genomics and
Right.
10x?
Right. Now, good question. So I don't think probably it's not gonna. For what I know, I don't think it's compatible with Flex, at least as of now. I would probably need to think about that if it would be compatible. I mean, this is probe based, so and we didn't include with this set of probe, we didn't include a bunch of genes that don't add any useful information success. There are no probes for ribosomal proteins genes. There are some probes for mitochondrial genes that we use for a QC purpose, for QC in the data essentially. And there are no genes for TCR. So that might not be the case for jump code for Flex. I think it's more relevant, definitely more relevant with 3' or 5'. Regarding your second question, I'm not sure actually what that really entails in terms of partnership. Yeah, I don't know. Maybe I can find more information and share it with you. If you have specific question, or if you want to know how to use their product with our library, I can definitely find more information. But it's not something that I've come across very often. Yeah. Yes. Oh, that's a good question. No. There are not, but we're gonna release a tech note. Don't ask me when, please, but if it happens before the end of 2022, I'll be extremely happy. It might be 2023 anyway. So we're gonna release this tech note in which we're gonna provide some guidelines or recommendation on how to spike in custom probes. But in terms of having a tool to design those probes, not that I know of. There is in the meantime, if there is interest in that, what I would say is let me know. I'll pass this information to our team. And there is also a paper in which the authors spiked in custom probes that is on Visium for FFPE. So it's not exactly single cell Flex, but the method is similar. It's similar enough that we can think about perhaps using that paper as a starting point, and then using the tech note as soon as that becomes available. Yeah.
First question.
If looking for rare cell population within a tissue sample, is it best to store the tissue in flex buffer and then dissociate later? Or perform the enrichment first?
Yeah.
and fix it?
It's a good question. So what I would say is it depends on, I would say depends on the sample, depends on the frequency of those rare cells. If we need to do FACS sorting because this cell population is very rare or sufficiently rare that we do need to do sorting, my recommendation will be to dissociate the tissue, sort the cells, and then if we have enough cells, fix the cells in the flex, but in the fixative. We don't recommend sorting fixed cells simply because we don't do that in-house. Not saying that is not possible, but we don't really have experience on that. I know that there are, at least BioLegend has some antibodies that they claim to work with fixed cells, but it will need to be tested. And so in that case, I would say it really depends on how many cells we are talking about, what the final number of cells is, what the frequency is. But one possible avenue would be dissociate the tissue, sort the cells. If we have enough cells, we do that and then we fix them. Yeah.
You went over the gene coverage earlier, but I do recall that it's been about 300 to 400 genes for the single cell RNA-seq. Do you have correlation of what the sensitivity would be to compare transcript numbers from bulk RNA-seq?
So I'm not sure about the 300 to 400 genes with the 3'. When that happens, typically is an indication that the sample was dead or dying, or anyway, the sample was compromised. We don't see only 300 genes per cells. That is an indication. That's an indication that the sample quality was poor, was bad. So for a typical healthy sample, we can see anything between 800 to 7,000, 8,000 genes, depending on the sample type, depending on sequencing depth. So that I would probably we need to talk. We need to have a conversation because I like to understand a little bit better why there were only 300 genes per cell, and because that definitely it's not a common scenario. And when that happens is because there is typically an issue with sample quality. Bulk RNA sequencing, no, I don't think we have any correlation with bulk RNA sequencing. It's hard to compare it because they're really two significantly different approaches. I don't think we typically compare, even with 3' chemistry or 5', we don't typically compare the number of genes that you detect with bulk and single cell. And once again, the number of genes that you detected with single cell depends on the type of sample and sequencing depth. So, but no, I don't believe we have any correlation with bulk RNA sequencing. I don't believe we run any bulk RNA sequencing. Sure. Yeah.
So right now we have the mouse, the humans, prophase approach for the flex. Do we have any plan to release other species?
Yeah. So good question. I'm not aware of anything in the pipeline, or that doesn't mean that there is no plan. It means that I don't have visibility, or I haven't been told any plan about releasing probes for, let's say, non-human primate. It can be rhesus macaque or rats. What I would say is, if there is interest in using flex on species other than human and mouse, and I've been having this conversation more than once with customers, it would be good if we could connect and see what species we would like to profile, And in the meantime, we pass the information to our internal team. And of course, there is always the avenue, the possibility of designing custom probes. That is something that can be done. We know the structure of the probes. We can pretty much share the structure of the probes. We cannot share the concentration of the probes, but that is where the optimization will start. Sure. All right. Yeah, if there are any questions, feel free to reach out to me. And as I said, I'll be more than happy to share the slide with you all.
Oh, yeah, yes, thank you.
Oh.
Yes, there's one more, so sorry.
Oh.
Thank you. So sorry, guys.
No worries.
Still, do you capture, if not multiplexing, the samples?
Oh, yeah. Sorry, I didn't mention that. Good point. So you can still capture up to 10,000 cells per channel or per sample when you multiplex it. It's 10,000, yeah. And yeah, I didn't mention that. So single plex samples, that would be 10,000 cells per sample. If you multiplex up to 4 samples together, that is still 10,000 cells per sample. If you multiplex 16 samples together, that goes down a little bit, is 8,000 cells per sample.
Okay, so thank you to 10X for coming today to give this seminar to tell us a little bit more about the technology that's evolving. And I would like to say, if you have any questions following this, please reach out to 10X and the Gene Expression Center. We'd be happy to set up a meeting with you to discuss your interest in the 10X platforms, whether it be on the single cell side with fresh or fixed, access to our new Chromium, not the new, to our older Chromium controller, independent access to that, what you need to do in order to be able to do that, or to learn more about the services that we have available. So thank you very much and happy holidays to everyone.